And many of you may know that lung cancer is globally important in the world. But in Asia, it takes more than 50% of the patient among all the cancer type. And we also have the issue of early onset. That means we have much younger patients, 10 to 15 years younger than the Western population. So this brings up the urgent issue to understand what is happening to the lung cancer in Asia. In Western population, the smoking is the most important factor. In Asia, the majority of the patients are never smokers. In particular, we have increasing female patients who are nearly never smokers. So we started a journey to study what happened to this never smoker by proteogenomics. In this project, we apply proteogenomics to analyze the tumor and adjacent normal tissue from the lung cancer patients. So by the very deep molecular profile, we discovered that somatic mutation profile is so different between our cohort and the cohort from TCGA, who are mostly smoking male patients. We also discovered two additional somatic mutational signature associated with nitrosamine, which is associated with the food preservation in the food industry, and another air pollutant related mutational signatures. And these two are unique in Taiwan. So this is the very first time that we use a molecular evidence to show that never smoking lung cancer is a distinct disease compared to the smoking lung cancer. Mass spectrometry is a super powerful tool to analyze protein and also the modification, which is beyond the measurement of genomic profile. So by using proteomics, we are able to dissect the cancer biology for our patients, to also identify the new subtype associated with the poor clinical outcome. And that is beyond the genomics tools. In the lung cancer study, we use proteomics to identify a new subtype called late-line subtype. That means the early stage patient who has the molecular profile similar to the patient from the late stage. A few years later, the follow-up data also verified that late-line subtype has much higher early recurrence rate. So that demonstrates that proteomics could be a fortune teller about the early recurrence of the early stage. And this patient should receive much more active treatment or more closer follow-up. So this demonstrates the power of proteomics to predict the patient's outcome. My lab has been working on clinical proteomics for many years. And plasma proteomics is such a new topic for us, which deliver much more user-friendly assay in the clinical community. However, we are just in the beginning of the plasma proteomics, and with such a high performance on the throughput, data acquisition speed, and the deep coverage. It's so exciting to see the new result from the arbitrary extra-room mass spectrometers. So now we have the confidence that plasma proteomics is ready for biomarker discovery all the way to validation. Proteins are the molecule to execute the function and also closely associated with initiation progression of the disease. In particular, most of the FDA-approved target therapy are targeting protein. So I believe that proteomics is the tool to provide the cancer biology and also to tell us what is happening at the patient level. And with the effort on proteomics research, I believe that it's going to be a scientific fortune teller to tell us the biomarker discovery about prevention, early detection, and the decision making during the patient's treatment.